Thanks, Daniel. I actually work at Austin Capital Data. I started this company in 2016, and we have a very particular mission. You guys are familiar with Justine's work. She's one of our data scientists, and I just want to. The talk's gonna be a little bit different. I think there's a lot of excellent, really highly technical work that's been shown. This talk is a bit more about what I see knowledge graphs and Grakn turning into in the future, and where they can have a very heavy influence. So you can see our slogan, data science for people everywhere. That's sort of the idea in a nutshell, that there's a lot of deep knowledge that's available, that's free, that we paid for, that we all own, but is not accessible. And that's not for any reason except you need to be an expert to understand a lot of this stuff. So I make the pitch that knowledge graphs are the way that people, like ordinary people, are going to enter into deep, nuanced understanding of scientific literature in particular, but other topics like cocktails as well, if you want to understand them at an expert level. So let's see if I can get my screen to respond. All right, so this is basically what I told you. We are largely an educational company. We do, a lot of teaching engagements, and we build knowledge graphs that are targeted for like everyday use cases. So the idea is that we make the bridge between highly technical information and things that people like other people who don't have access to that information really care about. And Grakn's one of our main allies in this. So this presentation is about one of our graphs, the cancer literature graph. I'm actually a biologist, so is Justine by training. So we care deeply about the PubMed data set and all the literature that's been put in there. That's one reason why we're building out the back end, the scientific back end for things like cocktails. And here is cancer data. So let's see. There are various problems with this type of data. Part of it is management, organization of that data. Like if you look at scientific literature, there's 26 million highly technical papers in the literature. Very hard to manage all that, even if you're an expert. And then what do you do once you have things organized, right? We can sort of use Google to get an idea of what's in the literature. But how do you analyze that once you find those answers? And the knowledge graph is the thing that we think is going to help get at that information. Up to this point, we've built out 4 knowledge graphs. I presented these in various forums, all of them. So you saw a cocktail graph, that's Justine's project. And the key idea here is there's a science drill down to cocktails. I'm going to tell you about cancer graph, and this interfaces with PubMed to get information about cancer for people who have cancer, or if your loved ones have cancer. What would you care about, and would you want to share in that public resource? And then let's see, collaborations. So this was one of our first graphs. We can use PubMed to think about who's collaborating with who, what do they know, what are the scientific communities. I think Michael talks a lot about this in the panel, which was really fun to hear about. And the idea is similar. If you have a question, you want to know who can answer it, you could look at their publication record, right? You can identify the highest publisher, but there's a ton more information if you just look at who's working with who, right? So that's the collaboration graph. And then there's Biopredictor, which was our first project. And this was when I was looking for a job in 2007 already. It was a long time ago. I was stymied by the number of biotech firms and what they were doing. Who would be the biotech firm if you're in the startup space that you would want to take a risk on and apply for a job with or invest in. So the idea behind Biopredictor was to organize the biotech startup space, and it turns out we could mask which one of those companies were going to IPO and figure that out using machine learning and graphs. So that's that. Now this is where my personal starting point comes from. You can see we care about this idea, but my sister got cancer. She was diagnosed with stage 4 cancer, pancreatic cancer. And she underwent aggressive chemotherapy. Now, I mean, this may resonate with some of you, maybe everybody, because when something like that happens to you, and you have this, like I say, I went to Princeton and studied genomics, and I have a degree in biology, and I run a data science company, and I've read, scientific literature, took a class on that involved, studying how cancer works and this kind of thing. But like, what do you actually do when something like this happens? Do you, dig into the literature and make your own recommendations? Do you trust your doctor? Like, how does that unfold in a person's life, even if you have the skills to have some understanding of what the actual science is? And my sister's not a scientist, so I wanted to see if I could sort of bridge that gap for her, just because it was something we're already doing, and it really should be possible. So she's actually a very popular person. She got a lot of contributions. You can see one of her posts over here. So you can see that there's a lot that you can do to support, but there's a piece that's missing, and that's where I wanted this project to begin. So you can read the literature, okay? Like I've heard a lot of people say this, okay, my father got cancer, my sister got cancer, and I went on a mission and I read all the literature, right? And I figured out the best case, the best practice, and I go to all the meetings. Okay, so that's good. That's really good. Your doctor probably has already done it. I mean, I'm not saying that that's a bad thing to do, but I think that sort of heroic effort should be, I guess, supplemented with better tools, right? Like, it shouldn't have to be that you've got to read the literature from the beginning every time, and then there's 105,000 pancreatic cancer papers. So even if you do your best, most likely you're not going to get a good picture of everything that's out there. You might see the highest profile papers and have an opinion, but I do think we can do better. So you can sort by relevance and develop an opinion, right? This is a mammoth task, right? Her doctor's probably already done that. There's a panel of experts who have opinions as well. So where can we leverage? So I actually went to her and I said, okay, what do you need? I mean, if I could build a graph, if I could give you information, what do you need? And she said, chemo is really hard, and it's really hard to stick out. And it turns out that a lot of people do really well in chemo until they stop doing chemo because they'd rather die than do more chemo. So that's where I dove in with this analysis. And the question is like, can we give somebody who doesn't have any training background in like what chemo side effects, what causes chemo side effects, and if there's anything out there that actually can alleviate them. So I set out to make a subgraph. Obviously, 105,000 papers is a lot, right? You can actually filter this down. So the tool I wrote, you can put a term in and query PubMed and get back a subset of papers, which to me is fine because 306 publications that are highly focused is probably plenty. So keywords, abstract text, author, chemicals, and again, keywords. This should be mesh terms, I think. We just extracted those very basic attributes and then put them into Grakn and built a graph from that using Biopython. I'll show you some of the Biopython code. I cut down a lot of the technical slides because I presented this to a Python group, but I know you guys really understand how to assemble a Grakn database, so I don't want to bore you with that. So this is just, the basic start, right? I made a key space PubMed ref cancer, and you can just see some part of the schema here. Let's see. Yeah, and I I logged all this stuff in batches, stored it in full files, right, as square tables because it's an API call. You can see that Entrez, right? So Biopython Entrez eSearch, you can pull directly from, you can pull paper metadata like the data I showed directly from the API, right? So you just have to parse it. That's how you do it. And then what you get back is the important part are the PubMed IDs here, right? So I can batch these PubMed IDs and then requery the database to get whatever I want, the very specific slice of data. And here's the code for that. And there's the list. Okay, and then, so here's the main wrapper. to partition the list and to just basically insert papers, right? Yeah, and you can see the fetch details method. So that's going back to the API to get the specific details. And then this is a loader that actually loads the data into the Grakn instance. Here's a look at what fetch details looks like. It's very much like the ntras poll. It's just another call to ntras, but it gets very specific information back. And then you can see what the data frame looks like, the ultimate data frame. So we load it into this data frame, and you can, we can verify that nursing implications of chemotherapy agents and their associated side effects, right? So we're hit with pancreatic cancer. So we're hitting the mark here, and then we've got some information. We've even got year information. So this facet, thanks Brett, is pretty interesting, right? To see how this evolves. Maybe even making a year-wise layered graph might be really cool. That you could explode and see how things evolved over time, because it should be going towards a specific direction, or maybe there are trends, right? Maybe different opinions are influencing the treatments over time, but that's a pretty cool idea to put in that time component. Okay, so here's just my parser. Again, running low on time. I don't want to bore you guys with that, but this is yeah, so there's actually, going back to the graph migrator that was shown, this was a challenge, parsing this stuff out, because sometimes these dictionaries are nested. They seem to change form and capitalization. I don't know if that's at PubMed, but sometimes I'll run this code and then I'll come back 3 months later and it doesn't work anymore. So you've got to put a lot of fail safes in there, because it seems as if those API objects are mutating somehow. So yeah, this is probably one of the most challenging parts of this. And I think everybody here recognizes how, how the data parsing is sometimes challenging. So you can see what the data frames are. At this point, it's very easy, right? Everything's nice square table, and you can get things to work pretty well. Okay, and then here's just an assertion statement. And again, I'll skip to the good stuff. Insert the data into the graph. Okay, yeah, we know this. Okay, so just back over here, what I'll say is I built this pancreatic cancer chemotherapy side effect graph, right? So you can see I've got these 306 publications that are highly focused for chemotherapy side effects. They've got these attributes. Important for the first analysis was chemicals, because I wanted to know what the chemotherapy agents are, and what we give somebody a lever, right? So 306 is big, but maybe it's too large to read all these things. So we have to sort of summarize this, right? How would you summarize this for a regular user to, in a sentence, give them some leverage so they can use this tool more often and get, educated stepwise. So the question might be, what are the most important treatments, right? Treatments we can get at by using chemicals. What are the most important ideas? Ideas we can get at by using keywords and mesh terms, which is another type of keyword that comes in with that. And then who can we connect with about these treatments? So this was another question that Tracy had. She said, are there people who are doing experimental treatments? Who are the best people in the world? You could probably judge that by maybe the number of publications that they have. So those things should come out of the graph as well, where we can query the institutes, list the institutes that are producing the most scientific work. Okay, so here's the query that I'm going to present. And again, in the interest of time, I'm going to skip. Let's just take a look at this. So this code allows us to ask basically any question in the PubMed literature, right? So in our case, it's a pancreatic cancer question, but with that query term, you can actually just make any small subgraph that you want that has to do with biology, right? So this question is, which pancreatic cancer papers have side effects listed as their topic or in their abstract, and then show us the chemicals mentioned in those papers, right? So we've got this P is a PubMed article, it has a title, it has PubMed ID and chemical, right? And then we've got this relationship here, ChemCont, right? So this is a PubMed chemical container object, and then we produce the paper and the chemical container output here. Yeah, and this is the same idea, right? What are the most important ideas and what are the most important chemicals? So here's the schema. Again, it's very basic. I think we, for this application, we don't need an incredibly elaborate schema, and this makes it very dynamic, right? So you've got author, mesh term, journal, chemical, PubMed article, and keyword, and we can get to the articles that are interesting, and maybe we'll start building up a reading library. Now, this is the chemo side effect subgraph. So when you do that query, you get this big hairball. So here's where the rubber really hits the ground, right? Like, even if I was looking at this, it would take me a while to figure out what's important here, right? This is kind of hard to look at. I mean, it looks beautiful, right? You can see there are some things that are not connected to the main body. There's some stuff over here. You see that structure starting to emerge, but really, how do you serve this up in a way that's palatable, that can give traction, right? Like, I know that a regular user is probably not going to spend a whole lot of time digging into this. They're going to move on to something that's easier to access. So how would we organize this? Well, it turns out that you can change the objects to different colors, right? So I can make chemical blue or purple, PubMed article red, and then we can start to see if we stretch these things out based on the number of highly connected chemicals, because that's what we're interested in, like what are the chemicals that are highly used. We can see that there's actually some very obvious structure, right? Once we get this into sort of a proper format. So there's these two nodes over here, or two chemical nodes over here, connected by a ton of papers. And then there's something out over here that's connected to those two as well, right? So what are those terms, and does it give us any insight that we could transmit to somebody who might want to learn about cancer in a hurry? So let's see. So we can see that this is gemcitabine, and that this is deoxycytidine, right? And so those are two chemicals that are used in the treatment of cancer, and they can actually, do a little Googling and figure out that, bingo, this is something that causes severe side effects, right? So this might be something that's very interesting to read about. There's a ton of stuff to read about it, right? It's just as simple as going now and clicking through these titles and then maybe saving them somewhere. And gemcitabine is a deoxycytidine analog, right? So look at the structure of these two things. It's really similar. So there's actually some interplay between those two items. And we know that this makes people really sick. It also makes them cured of cancer. So this is probably what she's complaining about. Although I don't know that because I haven't gone into a medical record, but I'd be willing to guess that this would be a good candidate of what the problem is. Let's see, antagonistic effects. It turns out that actually if you put in deoxycytidine and gemcitabine and pancreatic cancer into a search engine, you can find out that these things antagonize each other. Not a particularly surprising revelation. However, I think that this is a dive-in point, right? So for me, this is sort of mission accomplished, because once I present this stuff to her, she's going to start reading, right? So that's what we're going for. And it turns out that the deoxycytidine comes out of pancreatic stellate cells, which I'm assuming are overactive in cancer, and then it stops the effectiveness of this drug, right? So it reduces the effectiveness of this drug. So that's already interesting, and that's where we want to be. Okay, so what's next, right? Find the most important terms in this graph. Okay, so we found a bunch of terms. It turns out there's two other nodes that are kind of highly connected. This one is antimetabolites and actinoplastic, and then this one's antinoplastic agents. Turns out antinoplastic just means chemotherapy, so it's like a generic term. So I think this would be the most important revelation. However, if you want to do something like what she was asking me, she's like, okay, I know what my doctor's telling me. I want to hear about the stuff that nobody's talking about, right? So that's when you can spend some time poking around on these nodes over here, and maybe one of these is a bleeding edge treatment that, that we haven't talked about and your doctor hasn't read about because it's not in this cluster, right? Okay, so what do we do? What's the plan for this? Why do we do this? Why do we build Cocktail Graph, right? the plan is to open source these things, right? At least have a front end that's open source, that's coming, that's available, that people can use to sort of fall in love with this stuff, right? To just dig in and fall in love. Now, how do you make that useful, right? You could build like a bokeh display, which isn't in the plans, right? But it'd probably be cool to have like downloads of the data, right? So when somebody dives in, they can actually get to the data. The data source, like I showed you, the schema is very thin, which I think is a great jump starting point, because when all this stuff comes at an individual, it's very hard to assimilate that. But once you're in, right, then the data source should allow drill down, right? It should be able to go as deep as you want. And then maybe an update function and time course. That's the stuff I was thinking about when Brett was talking. That'd be great, you know, to have just this in layers, and then we can start talking about this in 4 dimensions, right? Or yeah, 4 dimensions. Time dimension would come out of the screen, right? And maybe there's some insights that are just not generally accessible to a human. All right, and then user concerns. I think that this is really, we spent a lot of time thinking about this. What does the user care about? What's going to make them like this stuff versus not like this stuff? Obviously, an intuitive front, that's the biggest deal maker or deal breaker. If somebody starts poking around and they're getting value, versus if they poke around and they get lost and fall asleep. Those are 2 very different pathways. The ability to drill down, right? Once you catch somebody, you want to keep them. So I think drill down is really important. This, I think, is huge, like memory. Like when somebody goes to the site, there should be some storage for their account, so they, you know, can go back to it and find the same papers. Obviously, this is a user interface issue that could make or break the system. And then for this, I presented this at Grakn Cosmos, and a number of people who are involved in cancer research said, you know, when are you going to make this available? So obviously, pancreatic cancer is a specialized use case, but you know, making this available for multiple cancer types is very easy, right? So we could have a drop down on the right, and you could switch to your cancer type of interest. So there it is. So again, take a look at this this schema over here. I'm going to steal from Grakn and show their COVID-19 graph. This is another biological graph, right? This BioGrakn. All the code of this is open source. To be very easy to put this on as the second level, something like this that pulls from all the public data sources and gets drill down, right? So either one of those chemotherapy agents, we could drill right down into all the other interactors, the genes that interact with it. This is a much richer schema, right? And you can see that, you know, there's sense to this, right? So the research entities, this would be an obvious way to find the departments that are doing work with that, right? You can see organization, laboratory, company, institution. These are publication items, right? So these are all publications. We could use that as an entry point. Patents, which we don't have in there if you're a VC, right? Or whatnot. This over here, these are biological entities. So all the genes and the proteins are organized in their interactions. So very easy to just sort of piggyback on this BioGrakn implementation and make something relatively rapid. So this is our team. We have a lot of great people who we work with and just try and bring this stuff into the light of day. So yeah, I think that's it. Yeah, that's the whole pitch.
Gunnar, bravo. Well done. Well done. Thank you so much, as always, for sharing your insights and your work with us. I know everybody is always rely on you to bring the work that's going to have the impact close to home, so to speak. So really appreciate that. A couple questions in here. Actually, there was a couple shout outs of maybe some suggestions for you. So I'll leave those for you to check out. Brett says your visualizations are crazy interesting, but the force graph layout doesn't necessarily do them justice. Consider grouping strategies, and he's willing to be hit up. RL suggests NetMiner as something interesting for your team. And then he also has a question. Speaking about memory, what is the required size of memory to serve the graph? Does Grakn support lazy load? And On the lazy load question, I believe the answer is yes. But if you drop that into Discord, I know the guys on the engineering team would be happy to answer that.
Yeah. So I mean, that graph that I showed is not. It does hang up a little bit when I try and render everything, but you know, I've got what 16 gigs of RAM, and it runs fine on that. I think 8 gigs of a RAM, not on a virtual server, but on a regular machine, can run that graph as well. So it's not really taxing, right? You could have a server at home and you could serve that.
Yep, totally. And thank you for that question. Conrad asks, have you tried to model relationships between chemicals using predicates between them?
What do you mean predicates, Conrad?
And Conrad, since you are one of our panelists, you might come off mute.
I meant actually, if chemicals appeared in any public in any article, so probably there are some, you know, they might be ordered in subject object predicate. So we might subject predicate object. So we might find in a text a predicate between them, which would be a verb. That might be pretty good relationship to model.
Yeah.
Approach in BioGrakn and COVID.
That makes sense. So yeah, we haven't done it. That's not actually NLP. It's just pulling the chemical list, which is like just very convenient. It's a list of chemicals that are submitted with each paper. But yeah, that would be great, especially if we're doing. I think there are probably people who have already implemented, like yourself, those items, right?
Yeah, and we could have a pretty good data source for it, data set. So these are already mapped. So we can find it. So I think that you can use that. Actually, I can share it with you. So these are already mapped from PubMed.
That'd be great. Yeah. Then I don't have to do any mapping, which is fine. I'd rather be on at the very end point, you know, if I can. Yeah.
I love
it. Is that the named entity recognition? No, that's a different project, right? I know that you guys use named entity recognition in BioGrakn.
We use the data set is just SAMMed. I don't know if you have heard about it. But
no, yeah. I'll email you though. Yeah.
Thanks.
Love it. Way to go, Conrad. Throwing some added value there. So since we are at the end of our time tonight, I do want to use the last couple minutes. If anybody wants to share their thoughts or a question with Gunnar, feel free to raise your hand, and I can bring you. You can come off of mute and say hello and ask any question to Gunnar you have. Otherwise, Gunnar, thank you again so much. I'm really excited to see both you and Justine and the progress that you make from here on, and have you come back to a future Grakn orbit and share along.
Fantastic, thank you. That'd be great. We love it. We love coming and talking to you guys.